So today I'm going to be talking to you about my team's research on improving beta-galactosidase for GM1 Gangliosidosis treatment. Um, so, so Codexis is a protein engineering company, and so we're focusing on engineering enzymes for a number of different applications, and my team works specifically on engineering these for therapeutics. So when we do this, we want to sort of set out on what are we actually engineering. So we want to use what patient needs, what are the enzyme needs, what is the disease
Recording in
needs
progress.
for this directed evolution or protein engineering. So what we want to do is we want to come up with a therapeutic blueprint. What would the optimal therapy look like? What kind of activity does the enzyme need? What type of stability? Is there an immunogenicity profile we need to look at? The developability and manufacturability, can we make it easier, cheaper? How are we going to make it? And then what's that modality or administration? How best could we take that enzyme or gene that we're engineering and deliver it? So we then start with that native protein. We then go through our CodeEvolver or directed evolution platform to develop that biotherapeutic that can hopefully get to the patient. So where did we start with GM1? So we looked at the early treatments of beta-gal. Looking into this, the enzyme is limited by the poor intrinsic stability and potency of that enzyme. So at neutral pH, it's relatively stable, loses about half its activity over a day. Yet if you put it in artificial cerebrospinal fluid into serum, you're losing more than 50% of its activity within 30 minutes, an hour. So what does that mean? Well, that means you're going to have much higher doses for gene therapy, could be potentially unsafe for patients in the dorsal root ganglion, ER stress, liver toxicity, depending on where that enzyme's going. It can also have a high cost of production. You're going to need more enzyme, more of that to go to the patient. Similarly, you're going to have to up that dose if you're using an ERT. That enzyme is going to have, going to be higher to produce. More of the enzyme is going to fall apart. It's not going to make it where it needs to go. And you're going to have that poor stability in the cerebrospinal fluid, in the serum, and get poor tissue distribution. So those fundamental properties of an enzyme that never really was supposed to leave the lysosome, that actually affects how it may be treated, how this treatment may be possible. So our goal was to use protein engineering to develop a superior GLB1 or beta-gal variants with these improved biophysical properties. And so how do we sort of do that in the laboratory? So we take the enzyme, we do libraries of variants of basically genetic mutations of beta-gal. These aren't pathogenic mutations. These are mutations that just vary the enzyme. We identify those. We then express each of those enzymes. We sequence it and test it for its activity, stability, tissue activity, all of the like. We then go back, mine that data, and then deconvolute those mutations. Which ones are beneficial? Which ones are deleterious? Which ones have no effect on the enzyme? And then we model those into the enzyme, and then we recombine that. And we do that iteratively over and over and over again, slightly making the enzyme slightly better each time we do this until we have this evolved beta-galactosidase variant. So this is what a sort of an evolution series looks like. The enzyme goes into the process in round one, goes through a few of these initial experiments of looking at expression, stability at neutral pH and biofluids. As we develop our assays, they start to get more complex as the enzyme increases its stability. So then we can look at cellular uptake in GM1 fibroblasts, thermal stability, and we start making better enzymes as we go along. And then we're able to look at more complex models of GM1 iPSC-derived neurons, removal of T-cell epitopes, other in silico modeling. And we made these different enzymes that we evaluated in a less high throughput, more structured environment. So I'll be highlighting the red and the green, BTX0204 and BTX0402. So what you can see right here is when we look at that serum stability, we completely flatten that curve, that loss of activity, where after 3 days or 72 hours, you can see there's absolutely no loss in the enzymatic activity here. The enzyme's completely stable. And really, after 24 hours, you can see you still have greater than 50% stability in artificial cerebrospinal fluid. And then when we take these enzymes, incubate them with patient fibroblasts, you can see greater than 6-fold of the enzyme is taken up inside of those cells and is active in those cells' lysosomes. So then we wanted to take this a bit more complex. This is in a test tube. This is also in a plate of cells. Can we do something a little more in a more complex neurological environment? So we took patient-derived fibroblasts. We reverted them into iPSC cells, so stem cells, and then we differentiated those into forebrain neurons. And you can see that somewhat in this picture here. We then also made neurospheres, so these are more complex organizations of those neurons where they form into these spheres. And then we did these dorsal forebrain cortical organoids. So those actually have neurons, glia, a number of the things that make it a brain. Some people call these mini brains or little things, so it's this very complex mixture of cells. We then packaged our enzyme into an AAV vector, either the wild type, that 02, or I'll call it 04 here for ease. And what you can see is that when these complex neuronal models were transduced, you could see 4 to 5-fold enzymatic activity in each of these models. So from this, we then wanted to see, so this is enzyme activity in these cells. We also looked at these cells to see what kind of the buildup of those metabolites of those biomarkers look like. And so when we looked at the neurospheres, you could see that the wild type have a relative level amount of GM1, all cells are supposed to, but in the knockout cells, you could see that there's an elevated amount in these organoids. When you add one of our engineered variants, it gets near to that baseline of where the normal cells are, and when you take that further engineered variant, you can actually decrease that even further. And so this is only after 72, so you can actually revert those GM1 cells back to that. And so then finally what we wanted to do was actually test this in an animal model, and these studies are still ongoing because these animal studies are very longitudinal. And so this is just the beginning of some of these data. And so what we did was we took the GLB1 knockout mice, we did a bilateral ICV injection, so take these vectors, inject it into the brain at a relatively low vector amount of 1.6 times 10 to the 10th genome copies per gram. And we looked at the wild type that, well, here it's called round 3, so that's 02 or 04. And what you can see is that when the wild type is transduced, you get it slightly above the knockout background. Again, that poor stability, that poor expression at only 8 weeks really isn't getting a large distribution throughout the mouse brain. But with the engineered variants, you can see the super physiological amount of the enzyme that's throughout the brain in these animals. Similarly, looking at a biomarker A2AG, the rest of the biomarker analysis is currently ongoing. You can see that, that 400 picograms per microgram increase in the knockout animals. When you add the wild type, you can see that it is reducing it down. It's bringing it down. But with the engineered variants, it's bringing it down almost to baseline there. And that's what's really exciting, is because with these engineered proteins, maybe at lower doses, at easier doses, we can actually start pushing these things lower in this disease. And so wrapping up, some of the summaries that we are continuing working experimentally on some of this in the preclinical setting, and that the properties of the engineered beta-galactosidase variants are better stability in biofluids, more stable at neutral pH, more activity in cells. We see efficient uptake in GM1-derived cells, and we see this high activity and reduction of A2AG in the mouse model. And again, we are continuing some of these in vitro and in vivo studies, getting the data from those. But last of all, I want to thank all of my colleagues at Codexis, specifically Oma and Charu, and Joseph Chen. They were pivotal in the GM1 program, driving it forward, doing the protein engineering, doing all the animal studies. And with that, thank you very much.